Thank you. Thank you to Dr. Topol, Ali, and the Scripps Institute for inviting me to talk a little bit about the realities of cancer screening. I will try not to be too bleak as I talk, but hopefully realistic. So, of course, why are we working on this? And Freenome's goal is to achieve early detection of cancer through blood. And you might have heard of some other companies that are working on this. And the reason for this is really, I think, summarized nicely by this slide, which shows the percentage survival over 5 years across the major cancer types that you might encounter. And what you see here is across all the cancer types, the difference between when you find a disease localized versus metastatic really makes a difference in terms of the outcome for the patient. And Freenome itself is currently focused specifically on colorectal cancer. And the reason for this is we actually don't know how to get a pan-cancer or a multi-cancer test into the clinic. And what I mean by that is it's not that we believe it's technologically infeasible, but we do believe that the challenges regarding showing utility for such tests, making sure that we do those sort of proper clinical studies, I think there are sort of insurmountable challenges, at least in current day, for us to really pull this off. Whereas colorectal cancer, it's nice in the sense that there is a very clear clinical path after you detect a disease early through colonoscopy, and the clinical trial design is relatively straightforward. So that's what we focus on. And I will talk a little bit about the challenges associated with it. And it starts actually as early as when you collect the blood. And a lot of different groups are collecting blood at different stages. For example, when screening is recommended, pre versus post-colonoscopy, pre versus post-surgery, pre versus post-treatment. And depending on which samples you collect and train on, you could actually cause your performance numbers to be confounded by various things that are not cancer-driven. So for example, if you're to collect samples post-colonoscopy, but the person still has the disease, most people might think, well, that's okay. The blood sample should have traces of cancer there. But the problem is when you collect the samples post a patient being told that they have cancer, the patient goes into stress response. And the stress response releases cortisol and other things that actually changes the degradation patterns of DNA, RNA, and proteins that are floating around in your blood. In fact, we actually saw this with a particular test called epiprocolon, where they first released numbers that were actually in the 90s in terms of sensitivity and specificity. And when they went to their pivotal study with intended use populations that were collected at the screening recommended stage, those performance numbers dropped to their 60s and 70s instead of 90s. And what happened there is their assay is sort of methylation-based. Methylation is particularly sensitive to stress response, and so they overfitted to a signal that was not necessarily disease-based. And then when they went and looked at the intended use population, of course, only a fraction of the signal was informative for cancer. So it does matter when you collect a sample, even at the sort of the training stage, so that you make sure you're fitting to the proper signal. Here's another example of how doing the wrong work can skew your results. This was actually from a couple of published works where we took their data and did a meta-analysis on them by building a classifier to tell the difference between cancer and healthy patients just using age and gender. And you can see here, this is probably the most extreme example of we took their cohorts and just built a classifier using age and gender, and it was able to distinguish cancer patients from healthy patients with 100% accuracy. And in this case, all of their cancer patients are actually older, systematically older than their healthy patients. And that could lead to all sorts of confounding of your results where you can't actually trust the numbers that are being put forth. Finally, just one last thing that I want to touch on that Freenome uses machine learning to sort of work on is normalization. So each one of these dots represent different institutions that we got samples from. And this is a t-SNE plot. It's basically a two-dimensional representation of the signal that we're capturing from the DNA. And in an ideal world, no matter which institution the sample came from, you should see this homogeneous cloud of samples, right? You should not be able to distinguish the signals based on which institution it came from. In this particular case, we do have an instance where especially the institutions, the red and blue dots here, are separate from the rest of the cohorts. And we essentially trained the learning engine to sort of understand this difference, and more importantly, how do we sort of normalize the counts of our DNA data so that you actually don't have that phenomena even before we start focusing on how do you actually detect cancer. In this case, this cohort actually systematically sent us more samples per day than this cohort. And the reason why that affected the results was that the institutions that sent us more samples per day had the samples sitting out for longer before they were actually processed. And so there was a little bit more degradation in these samples compared to these samples. So I'll talk a little bit about sort of debunking some assumptions of early cancer detection, which I'm sure many of you have heard about early detection and various efforts in there. The first assumption that I'd like to debunk is that there is enough signal in early cancer coming directly from the tumor that you can actually use for early detection. The second is that you only need a few of these signals, like a few mutations, to make an effective early detection test. Both are false. The first is just looking at the tumor fraction. If you're familiar with ctDNA, these are DNA fragments that have been spewed out by your cells as they apoptose or necrose into your bloodstream. And you find that the biggest challenge here is in stage 1 and stage 2 especially, the amount of DNA that you can actually detect that comes directly from the tumor is actually really low. So this is a little bit hard to see, but there is actually a blue bar and a gray bar here as well. And you can see that while some of them you can actually detect, even in stage 1 and stage 2, most of them have no detectable tumor fraction in the blood. And that causes a serious amount of problem when you're actually trying to capture these fragments to do detection, because the hypothesis is that if these DNA fragments are coming from the tumor, they would have driver mutations that you can actually detect from the blood. The problem is at this tumor fraction, you're talking about 0.01% or less is the tumor fraction. The amount of blood that you essentially have to collect to even get these fragments that are coming from the tumor into your sample so that you can actually detect it becomes prohibitive. So for example, if you're to collect 80 mils of blood per patient or 8 full tubes of blood, the best you can actually hope for is about 56% sensitivity based on sort of the calculations that you can do on tumor fraction of 0.01%. If you were to want to go, say, 95% sensitivity using primary tumor signatures, you're going to have to collect some like 300 to 350 mils of blood per patient, which is about 10% of the blood in your body. So I'm not going to belabor this. There's all sorts of math that goes into modeling this out. But the long and the short of it is you can actually figure this out by doing math. You collect 80 mils of blood per patient. That gives you about 32 mils of plasma, which is roughly 32 nanograms of cell-free DNA. That's about 10,000 genome equivalents. Okay, so if your allele frequency for your tumor allele is about 0.01%, that means of that 10,000, you have about 1 tumor equivalent that actually comes from the tumor, which means if you're to try to solve this problem, say with, let's say, 35x whole genome sequencing, that means you're trying to play this game of capturing that 1 out of 9,697 fragments in a particular location in the genome by sampling 35 times. I'm not going to go into the math of it, but it's really low, the likelihood of you succeeding. And that's assuming that you collected enough blood to actually capture any fragments at all. So, I mean, this is for a less sophisticated audience, but you can do this in terms of marbles for those of you that remember your probability class. But the main point here is you can obviously have these marbles across the entire genome. So the hope is that if there's enough mutations, then you can sample enough times across the genome to capture the signal of choice. But that doesn't really work either. So this actually uses the math that we just looked at and models this out, where it models out on the x-axis number of genomic events, whether it's copy number variations or mutations that you would essentially need to detect, and the sensitivity that you can achieve based on the sampling problem. And it turns out that number is much larger than what most people think about when they're thinking about creating a diagnostics test. About 1,400 is what you need for a high sensitivity test. And if you look at, say, all of TCGA and you're being really generous with statistical cutoffs on what a copy number variation is and things like this, you're talking about 300 copy number variations in late-stage colorectal cancer. So that's not enough signals there. Let's see what happens when you actually sequence much, much deeper. Again, this is assuming that you collected enough blood to capture the signal, but there's no doubt that you're going to actually capture the signal of choice. So say something like 60,000x. Obviously, the number of genomic events does go down that you need because the specificity of your call is much higher. But it's still about 30 or so signatures that you need. And if you look at some like TCGA, you're talking about 5 to 7 driver mutations that you can really detect consistently across patients. So even then, that's going to give you about 50% sensitivity max. So these are the reasons why this is a challenge. And I'll just jump to sort of what we believe the requirements are for effective cancer screening actually looks like. You need signatures that are coming outside of the tumor. You need secondary signatures. And we believe there's actually no single analyte solution for cancer screening because the signals that that's coming from the tumor is just too little. So you have to look at multiple analytes, but you also need to look at multiple analytes to increase specificity of each signal so that you can minimize the number of actual signals that you need to achieve early detection. So what do we mean by these secondary signatures? When you look at the tumor, the tumor is very small in an early stage, but there are other things that interact with the tumor. For example, tumor-associated immune cells, like macrophages that you can actually look at that are also turning over, and you can detect those signals. And then there's also a healthy tissue around the tumor that's actually getting displaced as the tumor is growing that you can actually detect. And these things, as they die, they spew things out into the blood that are DNA, RNA, and proteins, and you can actually capture this, for example, the immune system. And the cell turnover fraction is really what's assessed in the plasma because the cell has to die in order for it to actually spew out these DNA, RNA proteins. And that's a challenging problem. I'll skip over this. Because these are not intact cells. So you need to capture several elements of it to try to recapitulate what kind of cells are actually turning over in that given moment in your body that is reflective of whether you have a tumor or not. So I'm going to go over a few of these approaches, not all of the ones that we use, but a few of them just to give you a sense of how we think about this multi-analyte, multi-signal approach to cancer screening. The first one is really inferring gene expression from nucleosome footprinting. So most of the DNA that you actually capture in the blood are DNA that's actually guarded by nucleosomes or transcription factors or other protein complexes that are bound to the DNA. The DNA that's naked tends to get degraded very quickly by DNases. So ultimately, when you're doing cell-free DNA sequencing, you're almost doing a biologically inspired attack-seq to sort of see where the nucleosomes are sitting. And we showed this in a paper a couple years back where you get unexpressed genes, which have a lot of cell-free DNA that actually comes from that locus if you're looking at the transcriptional start site. Because these are genes that are turned off, largely sort of high density of nucleosomes over that region. And then housekeeping genes like GAPDH and beta-actin, you see this well-ordered nucleosome footprinting, and you capture overall less cell-free DNA because they're getting degraded faster because they're not protected by nucleosomes. You can compare this even to gene expression assays like RNA-seq, where the greater the FPKM, the less cell-free DNA you actually recover from the TSS. So in terms of doing machine learning to capture these types of signals, there's really two types, and I want to be sort of specific on how we do each one of them. One is to try to predict things like gene expression from the nucleosome footprinting. This is what I call a localized genome problem. There, you can look at all sorts of different genes across the genome. There's plenty of data for you to actually do this problem really well. The second problem, training a sort of model to actually differentiate between cancer and healthy patients with respect to sort of featurization, that is an untenable problem, at least in our hands, because there's just not enough patient samples for you to properly train a model to control for the high dimensionality space that you're actually dealing with in terms of just the number of genes, number of locations of the genome that you're looking at. So we supplement that step with a fair amount of data, both public and data that we generated. This is just one such flow where we take an entire genome as an input space. We whittled that down to sort of the transcriptional start sites here. And then you can do sort of additional filtering steps with additional data to finally get to a feature space that is more like 750 genes, 44 genes, to actually do this gene expression probability calculation. And of course, the underlying biology that we're banking on here is closed chromatin is tightly wound with dense nucleosomes, whereas the open chromatin tends to have far less nucleosomes in the region with transcriptional factors and other transcriptional machinery as part of the binding there. And you can read this out on things like V-plots here. So V-plots, what it does is this is genome location on the x-axis, so around the transcriptional start site, 500 bases on each side. And then on the y-axis is the fragment lengths of the DNA sequencing experiment that you're actually doing. So in this one plot, you can actually see not only nucleosome density, but you can actually see where there's fragments that have been protected by, for example, 2 nucleosomes. So this is a dinucleosome pattern that you're actually seeing. You can use these kinds of images to actually train your model. You can do V-plots as an input, and then you do a wavelet smoothing just to make sure that you're accounting for Gaussian noise. And then you can put it into something like logistic regression. And for each one of these transcriptional start sites, you can essentially infer whether it's a gene that's on or off. And it does pretty well when you're actually looking at this, right? So going from an easier task to a harder task, using these different methods of trying to capture the open versus closed chromatin. You have transcriptional start sites that are either constitutively on or off. I apologize, English is my second language. You have fairly high AUC performances here. And then you have things that are a little bit more granular in terms of the difference in FPKM, and the performance drops a little bit. And then most interestingly, you can start differentiating different cell types that are actually turning over, whether it's dendritic cells or monocytes, and the difference in the sort of peaks that you might encounter to infer the difference in gene expression of the 2 cell types. So a related work is obviously the DNA's hypersensitivity sites, which is part of what nucleosome footprinting shows. But again, this is where it's interesting when you're looking at cell-free DNA. It's actually inverse of the signal that you would get, for example, in an ATAC-seq experiment, where you are directly assaying the nucleosome binding. And I'm not going to take too much time on this, but again, there's a statistical way of looking at this where essentially the subset of DHSs that you're wanting to look at are the ones that are what's called medium correlation across the different patients. So if each one of these lines are different patients and the purple line is the average, you're really looking for things that are medium correlation as opposed to high or low correlation, because low correlation basically means there's nucleosomes everywhere, no matter what cell type it is. And then high correlation means that there's no nucleosomes in all cell types. And if you're trying to look for differences between cancer and healthy patients, you're going to do a better job filtering for this medium correlation signal. And again, there's ways to introduce other data sets like a reference monocyte DNA-seq experiment to start whittling down your feature space so that you're not doing this top-down machine learning problem that you don't have enough samples for. And then you can obviously train a retrospective samples and then test on prospective samples to see if your performance actually holds up. I'm not going to go over this for the sake of time, but there's multiple ways of doing cross-validation that we actually report in our papers, because you need to control for age and gender and all those confounders that we actually talked about before. Just looking at it from sort of a single analyte perspective, it's interesting because already these immune-driven signatures or extracellular or extra tumor signatures that we're capturing gives you that higher sensitivity compared to some of our competitors that are looking at this, that are looking at primary tumor signatures. You can see, especially for stage 1, some of the groups are reporting numbers between, say, 40 and 50% sensitivity. We're more in the 80% sensitivity just by capturing this immune signatures that tends to be amplified above tumor. And it's not something that's just weird with our samples, right? This is our signal looking at the immune-driven signatures. And in the same patients, we looked at tumor-driven signatures and what kind of performance that gives us. This is actually in line with what's been reported in literature before. Some people get nervous when we talk about immune signals and whether that's sort of specific to the tumor of interest or not. So we actually specifically train for those DHSs, for those gene expression profiles that are specific to the immune response of the disease of interest. So you can see here we tested GI, inflammatory, as well as other sort of controlled comorbidities that are down here. And you actually have high specificity against those other indications as well. I think I'm going to skip over this section just in the interest of time. I'm sorry, I had way too many slides. But the last thing that we really looked at was putting it all together. How do you sort of break down the different cell types? Because whether it's comparing to ataxic experiments, whether it's comparing gene expression profiles of the various cell types, it's kind of interesting to see that in healthy patients, you're getting most of that hematopoietic turnover, right? That's what's turning over here. But then we started looking at other cell types as well, and these are lung cancer samples, colorectal cancer samples, melanoma samples. And what you find here is in the cancer samples, it's actually the primary tissue that's lighting up, not the actual cancer experiment that's associated with it. So like if it's lung cancer, it's really, what it's saying is the lung tissue is actually turning over. In the case of colorectal cancer, it's the colon tissue that's turning over. And it's not that these are cancer cells. These are actually healthy cells around the tumor that's actually getting displaced by the tumor as it's dying, which is another amplified signal that actually gives us more specificity than just looking at immune-driven signals. This is just a short story about as you're merging different analytes and merging different features, there's a lot of different ways of doing this from a machine learning perspective, and how you do it does matter. And we came up with a method of doing this called where we measure the probability correlation metric between different analytes. So if two analytes have really high PCM, that means they're very correlated, which means they're much more likely to add noise than they're to add signal because they're contributing the same signal. So we're really trying to get at the small PCM, and that's exactly what this experiment shows is actually the AUC performance is lower than just looking at single analyte. The single analyte performance was about 0.92. If you add a bunch of features arbitrarily, you're actually going to get a lower performance. And if you focus on sort of the small PCM, then you get a higher performance. And you can test this along our patient cohort here. In this case, there's about 1,000 patients here that we tested this on. In the blue bar is the single analyte. Green bar is the small PCM analytes. And then the red is the large PCM analytes. And here's the sensitivities in stage 1 and stage 2. And you can see that the small PCM gives you the best performance, which far surpasses some of the other sensitivity numbers that you might have seen. So the next steps for us, we do have this clinical trial that's out right now, AI Emerge, where we're collecting about 3,000 patients. Only about 1,000 of those patients are intended use population. We're also collecting some not intended use population to compare. But that should be reading out hopefully later this year, after which we will do an FDA pre-submission and kick off our pivotal study next year. And we collect many sites. So with that, I'd like to thank my team. There's a lot more people here, but these are some of the leads of the projects that you just saw. And this is a very non-exhaustive list of people that worked on these particular projects within my company. And there's many, many more that I don't name here, which I very much apologize for. And thank you so much to some of our advisory board members for actually guiding us on not only indication choice, but also some of our science. Thank you.